Okay, let's unpack this. Imagine your body performing a high wire act. It's constantly balancing, adjusting, keeping everything steady.
Yeah.
Especially when it comes to energy, right? Managing where the fuel comes from, where it goes.
Yeah, exactly. And there are these incredible biological systems working behind the scenes, maintaining this delicate metabolic tightrope walk.
And right at the center, orchestrating this whole metabolic balance, is an organ we often think of for digestion mostly, the pancreas.
But the pancreas has this remarkable double life. It secretes digestive juices, absolutely, but it also houses these specialized clusters of cells, an endocrine powerhouse, really.
Releasing vital hormones directly into the bloodstream.
That's it.
Right, and that's where our deep dive takes us today. We're focusing intensely on those endocrine clusters, those tiny islands, the islets of Langerhans, and specifically the two absolutely fundamental hormones they produce, insulin and glucagon.
These are the primary conductors, you could say, of how our bodies handle the main energy currencies, glucose, fat, and protein.
Now, the material mentions other hormones too, amylin, somatostatin.
It does, yeah. Amylin, somatostatin, pancreatic polypeptide. But our mission today is really to go deep into the well-established crucial roles of insulin and glucagon.
And critically, to understand in detail what happens when that delicate balance they maintain breaks down, which leads to diabetes mellitus.
Exactly. That breakdown is key.
You've given us a stack of sources, research notes, and that's our map for this deep dive. We're going to try and cut through the complexity, extract the essential knowledge, get straight to the heart of how this system works.
Think of this as a deep dive crafted specifically for you, the listener, designed to give you the insights and understanding you need to really grasp this topic.
To master this information, really get it.
Yeah, like an elite professional navigating this metabolic terrain thoroughly, clearly.
Okay, so let's start at the beginning then. The physiological anatomy of the pancreas itself. Where are these vital hormones actually made? What does the sources say?
Well, the pancreas is described as having two distinct types of tissue. First, you've got the acini.
Acini.
These cells are responsible for the exocrine function, secreting digestive enzymes into ducts. Those lead to the small intestine to break down food. The material mentions this digestive role is covered elsewhere.
So that's one part, handling digestion. But the part we're focused on is the endocrine side.
Exactly. The second main tissue type is the islets of Langerhans. These are the endocrine core, the hormone factories.
Tiny islands within the pancreas.
Tiny, yeah. But millions of them. A typical human pancreas contains maybe 1 to 2 million of these islets. Each one is quite small, roughly 0.3 millimeters across.
Wow, that's a lot of them.
It is, and they are strategically organized around small capillaries. This setup is key.
Why is that so important?
Because it means the hormones secreted by the islet cells go directly into the blood flowing through these capillaries. Immediate access to the general circulation.
Perfectly positioned for rapid communication throughout the body. And these islets, they're not just one type of cell, right? They contain different populations.
We do. The source material highlights 3 major cell types, mainly distinguished by how they look microscopically: alpha, beta, and delta cells.
Okay, alpha, beta, delta.
Beta cells are the most abundant, making up about 60% of the cells within the islets. They're usually found in the central part of the islet.
Yeah,
They're the source of both insulin and amylin. The material notes that amylin is secreted along with insulin, though its precise physiological function isn't quite as thoroughly understood as insulin's.
Okay, beta cells. Insulin and amylin. What are the alpha cells?
Alpha cells make up about 25% of the islet cells, and they secrete glucagon.
The counter hormone to insulin, basically.
In many ways, yes. Then the delta cells, they're about 10%, and they're responsible for secreting somatostatin.
Somatostatin, okay.
And the material also mentions a smaller number of PP cells that secrete pancreatic polypeptide.
What's fascinating, though, is that these different cell types within an islet aren't just like roommates. They actually interact directly.
This local interaction is incredibly important for fine-tuning hormone release. The source points out these close interrelations allow for direct cell-to-cell communication within the islet.
So what one cell releases affects its neighbors.
Precisely. It means the secretion of one hormone can directly influence the secretion of another, right there at the source, before it even gets into the main bloodstream.
Can you give us an example of that local control?
Sure. The material gives specific examples. Insulin, for instance, is known to inhibit the secretion of glucagon from the alpha cells.
Okay, so insulin dampens glucagon release.
Right, and amylin, secreted with insulin, seems to inhibit insulin secretion itself, a sort of local feedback.
Interesting. And somatostatin.
Somatostatin from the delta cells has a broader inhibitory effect. It suppresses the secretion of both insulin and glucagon within the islet.
Wow, so it's like a local break on both major players.
Exactly. This internal communication system adds a really significant layer of regulation to how these hormones are ultimately released into the blood.
So the architecture of the islet isn't just a collection of cells, but a mini endocrine system with its own internal chat going on.
That's a great way to put it. It's a prime example of paracrine signaling hormones acting on nearby cells.
All right, let's shift our focus now specifically to insulin. This hormone has quite a history. The source material starts by acknowledging its discovery.
Oh, absolutely. The isolation of insulin back in 1922 by Banting and Best was truly revolutionary. The source rightly calls it life-saving.
Before insulin therapy, things were grim for severe diabetes.
Very grim. A diagnosis of severe diabetes mellitus meant rapidly worsening health, often death within a pretty short time. Insulin just dramatically changed that prognosis. It offered immediate, effective treatment.
Insulin is often simplified. People call it the blood sugar hormone.
Yeah.
But the source makes it clear its role is much, much broader.
That's a crucial point to grasp if you really want to master this. While insulin's impact on carbohydrate metabolism is profound, yes, its effects on fat and protein metabolism are equally, maybe even more critical, especially long-term.
How so?
Well, the source emphasizes that abnormalities in fat and protein metabolism due to insulin dysfunction are major contributors to the illness and death associated with diabetes. Untreated diabetes compromises the body's ability to build proteins, leading to tissue wasting, functional problems.
Yeah.
It's systemic.
So understanding insulin means understanding its effects across all the macronutrients.
Definitely. And the fundamental principle behind insulin's action, as described in the source, is linked to states of, well, energy abundance.
Energy abundance, like after a meal.
Precisely. Insulin is the hormone that signals plenty. When you eat, especially carbs, and your body has an abundance of energy available, insulin secretion ramps up. Its core job, then, is to promote the storage of that excess energy.
Okay, so how does it manage that storage across the different fuel types? Carbs first.
For carbohydrates, insulin directs them to be stored as glycogen, primarily in the liver and muscles.
Glycogen, the stored form of glucose.
Right. And if there are more carbohydrates than can be stored as glycogen, insulin stimulates their conversion into fatty acids. These then get packaged into triglycerides and stored in adipose tissue, fat tissue.
So excess carbs become fat under insulin's direction.
Essentially, yes. For proteins, insulin promotes cells taking up amino acids, the building blocks, and it encourages their assembly into new proteins.
Building muscle and other tissues.
And it also acts to prevent the breakdown of existing proteins. So when energy is abundant, insulin is the signal. Store it as glycogen, store it as fat, build protein, build up those reserves.
It's the body's chief storage manager. Let's look closer at the hormone itself, its chemistry, how it's put together in those beta cells. The source goes into some detail.
Okay. Human insulin is a relatively small protein, a polypeptide. Molecular weight around 500-808. It consists of two amino acid chains, an A chain and a B chain, linked together by disulfide bonds.
Those bonds are important.
Very. The source notes that breaking these disulfide linkages renders insulin inactive. They're essential for its structure and function.
How is this molecule actually synthesized inside the beta cell? What's the process?
It follows the standard pathway for protein hormones, but with some specific processing steps. It starts with the genetic code for insulin being transcribed into messenger RNA.
Right, the blueprint.
That mRNA is then translated on ribosomes attached to the endoplasmic reticulum, producing a larger precursor molecule called pre-proinsulin.
Pre-proinsulin, the initial product.
Yes. And within the endoplasmic reticulum, this pre-proinsulin is quickly cleaved, snipped, to form proinsulin.
Okay, so pre-proinsulin becomes proinsulin. Still a precursor.
Still a precursor, yes. Proinsulin is larger than active insulin, around 9,000 molecular weight. It contains 3 peptide chains, the A chain, the B chain, and a connecting peptide, known as the C chain or C peptide.
A, B, and C chains. Got it. What happens next?
Most of this proinsulin is then shuttled off to the Golgi apparatus, another part of the cell's machinery.
The processing and packaging center.
Exactly. In the Golgi, it undergoes further enzymatic cleavage. This is where the C-peptide is cut away.
Ah, so the C chain is removed.
Right, leaving the A and B chains linked by those crucial disulfide bonds, and that is the active insulin molecule.
So insulin and the C-peptide are generated together from proinsulin in the Golgi, and they're stored together.
Yes, the source states that both the finished insulin and the cleaved C-peptide are packaged together into secretory granules within the beta cell.
Ready for release.
Ready for release. And when the beta cell gets the signal to secrete, both insulin and C-peptide are released into the bloodstream in equimolar amounts. Equal molecular quantities.
Equal amounts. Does any proinsulin get released?
A small amount, yeah. About 5% to 10% of the secreted product is still in the proinsulin form.
And the functional activity. Is proinsulin or C-peptide active like insulin?
The source is clear. Neither proinsulin nor C-peptide has any significant insulin activity themselves. Virtually none.
So there's your byproducts.
Well, not necessarily for C-peptide. The source mentions something interesting there. C-peptide seems to bind to a membrane structure, likely a G protein-coupled receptor.
Okay.
And it activates specific enzyme systems inside the cell. Things like the sodium-potassium pump, endothelial nitric oxide synthase.
So it does do something.
It appears to. Now, the source stresses that the actual importance of C-peptide in regulating these enzymes physiologically is still uncertain, still under investigation, but it suggests the C-peptide might have its own biological roles. It's not just inert.
That's fascinating. Not just a leftover piece.
Yeah.
And you mentioned C-peptide has a diagnostic use.
Yes, this is a key clinical application. Measuring C-peptide levels in the blood is very useful, especially in patients with diabetes who are being treated with insulin injections.
Why is that?
Because the insulin they inject doesn't contain C-peptide. So the amount of C-peptide in their blood reflects how much insulin their own pancreas is still managing to produce.
Ah, it measures their natural production.
Exactly. In type 1 diabetes, where the beta cells are largely destroyed, C-peptide levels are typically very low or even undetectable. This confirms that the body's own insulin production is minimal or gone.
That gives doctors a clear window into the patient's remaining beta cell function. Okay. Once insulin is secreted into the blood, how does it circulate? How long does it stick around?
The source explains that insulin circulates almost entirely unbound. It doesn't latch onto plasma proteins much. However, it has a very short half-life in the plasma.
How short?
Average of only about 6 minutes. Within 10 to 15 minutes, most of it is cleared from the circulation.
Wow, that's remarkably fast. Why such rapid clearance? Why not hang around longer?
This rapid turnover is essential for the tight control insulin exerts. The body needs to be able to quickly turn off insulin's effects when blood sugar drops, for example. Allows for dynamic adjustments.
So it needs to be cleared quickly to allow for changes. How is it cleared?
Insulin that doesn't bind to a receptor is primarily degraded by an enzyme called insulinase.
Insulinase. Makes sense.
The liver is the main site for this degradation. It clears about 60% of the insulin from the blood as it flows through the portal vein, coming directly from the pancreas. The kidneys, muscles, and other tissues also help clear the rest of the circulating insulin.
So synthesized, processed, secreted, circulates briefly, and then quickly removed. A very dynamic life cycle. Now, how does insulin actually talk to its target cells? How does it deliver its message?
Insulin's message is received by the insulin receptor. The source describes this as a large protein embedded in the cell membrane. It's made of 4 subunits held together.
Curved parts?
Yeah, 2 alpha subunits located on the outside of the cell membrane, that's where insulin binds, and 2 beta subunits that actually penetrate through the membrane and extend into the cell's interior.
So the alpha parts are the docking station on the outside, and the beta parts stick inside the cell.
Exactly. When insulin binds to those alpha subunits on the outside, it causes a change in the receptor shape. This change triggers an enzyme activity within the beta subunits on the inside of the cell. It's a process called autophosphorylation.
Autophosphorylation. The receptor phosphorylates itself.
Yes. This activated beta subunit acts as a tyrosine kinase, meaning it can add phosphate groups onto specific tyrosine amino acids on other proteins inside the cell.
So the receptor itself, once insulin binds, becomes an active enzyme, starting a chain reaction inside.
Precisely. This activated tyrosine kinase then phosphorylates a whole cascade of other intracellular enzymes and signaling molecules. A key group mentioned are the insulin receptor substrates, or IRS proteins.
IRS proteins.
Right, and this phosphorylation cascade is like flipping switches, activating some enzymes, inactivating others. It effectively reprograms the cell's metabolic machinery according to insulin's instructions.
And the source breaks down the effects of this reprogramming based on how quickly they happen, right?
Yes, it gives a timeline. The fastest effects occur within seconds of insulin binding.
Seconds. What happens that fast?
The most dramatic is a marked increase in glucose uptake by about 80% of the body's cells, especially muscle cells and adipose or fat cells.
80% of cells start taking up more glucose almost instantly.
Almost instantly. But the source is careful to note. This rapid increase in glucose transport does not happen in most neurons in the brain.
Ah, right. We touched on that. Why are brain cells different again?
The source explains that most brain cells are already permeable to glucose, even without insulin. Their glucose uptake is insulin independent. This is critical because the brain relies almost exclusively on glucose for energy. It needs constant access.
So it doesn't need insulin's key to get glucose, but muscle and fat cells do. How does insulin unlock the door for them so quickly?
It triggers the movement, the translocation of intracellular vesicles. These tiny bubbles inside the cell contain glucose transport proteins, specifically a type called GLUT4.
GLUT4 transporters, they're stored inside.
Yes, they're normally waiting inside these vesicles. When insulin binds its receptor, the signaling cascade tells these vesicles to move to the cell membrane and fuse with it.
Popping the transporters onto the surface.
Exactly. Inserting the GLUT4 transporters into the membrane. This dramatically increases the number of open doors for glucose to enter the cell.
And this is reversible.
Very quickly. When insulin levels drop, these transporters are pulled back inside the cell within a few minutes, stored in vesicles again. It's a brilliant system for rapid, on-demand glucose uptake control.
A very elegant mechanism. What other effects happen within seconds?
The source also mentions that within seconds, insulin increases the cell membrane's permeability to many amino acids and also to potassium and phosphate ions, boosting their transport into the cell as well.
Okay, and slightly slower effects, taking minutes.
Within about 10 to 15 minutes, you see more widespread changes in the activity levels of many other intracellular metabolic enzymes. These changes are largely due to alterations in their phosphorylation state, kicked off by that initial signaling cascade from the receptor.
And then there are even longer-term effects, over hours and days.
Yes, these are the slowest effects and involve changes at the level of gene expression and protein synthesis. Insulin can alter the rate at which messenger RNA is translated into new proteins at the ribosomes.
Building new machinery.
Right. And over longer periods, it can even change the rate of DNA transcription in the nucleus, leading to increased production of RNA and consequently more protein synthesis, especially enzymes involved in energy storage pathways. The source says this effectively remolds the cell's enzymatic machinery over time, gearing it towards storage and utilization of nutrients.
So insulin's influence stretches from immediate transport changes right up to fundamentally reshaping cellular capabilities over days. A true master regulator. Let's focus now specifically on its effects on carbohydrate metabolism, perhaps its most famous role.
Okay. Right after you eat a high-carb meal, glucose gets absorbed into the blood. This triggers a strong surge in insulin secretion. And that high insulin level leads to the rapid uptake, storage, and use of that glucose, especially by muscles, adipose tissue, and the liver.
Let's start with muscle. How does insulin impact glucose uptake and metabolism there?
Well, during fasting or between meals, when insulin levels are low, resting muscle actually relies primarily on fatty acids for energy. The muscle cell membrane isn't very permeable to glucose without insulin's help.
Okay, so normally it burns fat. When does it use glucose?
Muscles become major glucose users under two main conditions. One, during moderate to heavy exercise. Muscle contraction itself helps GLUT4 transporters move to the surface, so high insulin isn't strictly needed then.
Exercise opens the door too.
Right. The second condition is for several hours after a meal. That's when you have both high blood glucose and high insulin levels. This combination drives rapid glucose transport into the resting muscle cells. And in this state, the muscles preferentially use glucose over fatty acids for energy.
So insulin helps resting muscle grab that post-meal glucose. And if the muscle isn't exercising, what happens to that glucose?
If the muscle isn't active, the abundant glucose entering the cells under insulin's influence gets stored as muscle glycogen.
Building up that internal fuel reserve.
Exactly. Muscle glycogen can reach concentrations of maybe 2% to 3% of the muscle mass. It's a readily available energy source for the muscle itself, particularly useful for bursts of activity or anaerobic metabolism. The source mentions data showing insulin can increase the rate of glucose transport into resting muscle cells by at least 15-fold. It's a huge effect.
15 times faster uptake. Wow. Okay, beyond muscle, the liver is absolutely crucial for glucose management. The source calls insulin's effect here one of its most vital after a meal.
Absolutely. Insulin causes most of the glucose absorbed from a meal to be rapidly stored in the liver as glycogen. This is a key buffering function.
Buffering, how so?
It soaks up the glucose surge after eating. Then, when blood glucose starts to fall between meals, the liver releases this stored glucose back into the blood to prevent levels from dropping too low. It smooths out the peaks and troughs.
Okay, how does insulin trigger this storage in the liver? The source mentions several simultaneous actions.
Right. It orchestrates a few things at once. First, insulin inactivates the main enzyme in the liver that breaks down glycogen, liver phosphorylase. So it prevents the stored glucose from being released.
Stops the GUYTON.
Second, it increases the activity of an enzyme called glucokinase. This enzyme phosphorylates glucose as soon as it enters the liver cell, essentially trapping it inside because phosphorylated glucose can't easily leave.
Traps it inside. Got it.
And third, it increases the activity of the enzymes that actually build glycogen molecules from glucose, especially glycogen synthase. So it ramps up the storage process itself.
So simultaneously, shuts down breakdown, enhances trapping, and promotes synthesis.
Yeah.
Very efficient. How much glucose can the liver actually store as glycogen?
It can store a significant amount. The liver can increase its glycogen content up to about 5% to 6% of its total mass. That equates to roughly 100 grams of glycogen in an average adult liver.
100 grams. That's a decent reserve. And the release phase between meals, how does that reversal work?
When blood glucose begins to fall, insulin secretion rapidly decreases. This drop in insulin quickly reverses all those storage-promoting processes.
The signals change.
Exactly. Glycogen synthesis stops, liver glucose uptake slows right down. Critically, the enzyme for glycogen breakdown, phosphorylase, which was inhibited by insulin, becomes active again. It starts splitting glycogen back into glucose phosphate.
Okay, releasing the units. But it's still phosphorylated.
Right. So the final step is another enzyme, glucose phosphatase, which was suppressed by insulin, becomes active. It snips off that phosphate group. Now you have free glucose, which can diffuse out of the liver cell and back into the bloodstream to raise levels.
So the liver acts as this dynamic buffer, soaking up glucose after a meal, releasing it between meals, smoothing out those blood glucose fluctuations.
Precisely. The source notes that severe liver disease really messes this up. It severely impairs this buffering capacity, making it much harder to maintain stable blood glucose.
What happens if the glucose load from a meal is just massive, more than the liver can store as glycogen?
Yeah, if more glucose enters the liver than it can store as glycogen or use for its own immediate energy needs, insulin strongly promotes the conversion of that excess glucose into fatty acids.
That carb-to-fat conversion we mentioned earlier.
That's the one. These fatty acids are then assembled into triglycerides and packaged into particles called very low-density lipoproteins, or VLDL.
VLDL, like little fat delivery trucks.
Pretty much. They're released from the liver into the blood and transported to adipose tissue for long-term storage as fat. The source explains this involves breaking glucose down to acetyl-CoA, the main building block for fatty acids.
So excess carbs are efficiently converted and shipped off to fat storage under insulin's management. Insulin also stops the liver from making new glucose from scratch, right? Gluconeogenesis.
Yes, it inhibits gluconeogenesis.
Yeah.
This is the process where the liver makes glucose from non-carb sources like amino acids or glycerol. Insulin suppresses this pathway.
How does it do that?
Partly by reducing the activity of the key liver enzymes needed for gluconeogenesis, and also partly by reducing the availability of the precursors, especially amino acids released from muscles. Insulin helps keep those amino acids locked up in protein stores.
Conserving protein, stopping unnecessary glucose production. Makes sense. We mentioned earlier that brain glucose uptake is different. It doesn't need insulin. The source really emphasizes why this is so vital.
It's absolutely critical. Unlike muscle and fat cells, most brain cells can readily take up glucose without insulin. And the brain is unique. It primarily relies on glucose for energy, almost exclusively under normal conditions.
So it needs a constant, reliable supply.
Exactly. Therefore, maintaining blood glucose levels above a certain minimum threshold is vital for normal brain function.
What level is that critical threshold? And what happens if it drops below?
The source states it's essential to keep blood glucose above roughly 20 to 50 milligrams per 100 milliliters. If it falls below this, the brain is starved of energy. This leads to the symptoms of hypoglycemic shock.
Hypoglycemic shock. What does that look like?
Initially, nervous irritability, trembling, sweating, then potentially fainting, seizures, and eventually, if it persists or gets worse, coma. This really highlights why the body's glucose control system is so fiercely protective. It's safeguarding the brain's fuel supply.
Okay. And insulin's effect on other body cells, besides muscle, liver, and brain.
In most other cells in the body, insulin also increases glucose transport and utilization, similarly to its effect in muscle.
So most tissues become more glucose hungry with insulin.
Yes. And in fat cells, this transported glucose plays another important role. It provides the glycerol backbone, specifically alpha-glycerol phosphate, needed to assemble fatty acids into triglycerides for storage. So insulin indirectly promotes fat deposition this way too, alongside its other direct effects on fat metabolism.
So glucose uptake in fat cells helps lock away the fat.
Oh.
Okay, we've covered carbs. Let's dive into insulin's equally important effects on fat metabolism.
Yeah, and the source really emphasizes the long-term importance here. Insulin deficiency is a major driver of severe atherosclerosis, hardening of the arteries. That's a primary cause of heart attacks, strokes, and other vascular diseases in people with poorly controlled diabetes.
So problems with insulin's fat effects have serious long-term consequences. How does insulin normally promote fat synthesis and storage?
Well, first, insulin acts as a fat sparer. By increasing glucose utilization all over the body, it automatically reduces the need to burn fat for energy, so less fat is broken down.
Spares the fat stores.
Right. It also directly promotes the synthesis of fatty acids, particularly from any excess carbohydrates, mainly happening in the liver.
We talked about that VLDL pathway.
Exactly. Those fatty acids are packaged into triglycerides, put into VLDL particles, and shipped out to the adipose tissue. But insulin plays another direct role there too.
At the fat tissue itself.
Yes. It activates an enzyme called lipoprotein lipase, which sits on the walls of the capillaries in adipose tissue. This enzyme breaks down the triglycerides carried by the VLDL and other lipoproteins,
Releasing the fatty acids so they can enter the fat cells.
Precisely. It unpacks the delivery truck, allowing the fatty acids to get inside the adipose cells.
Okay, so it helps package carbs into fat in the liver, ships them out, and then helps unpack them at the fat cell doorstep. What else does it do inside the fat cell to promote storage?
Once fatty acids are inside the fat cell, insulin has two more crucial effects. First, it strongly inhibits an enzyme called hormone-sensitive lipase.
Hormone-sensitive lipase, what does that do?
That's the main enzyme inside the fat cell responsible for breaking down stored triglycerides back into fatty acids and glycerol, allowing them to be released. By blocking this enzyme, insulin effectively locks the door from the inside, preventing stored fat from escaping back into the blood.
Prevents fat mobilization.
Yeah.
Okay, what's the second effect?
Second, insulin promotes glucose transport into the fat cell, which we mentioned provides that essential alpha-glycerol phosphate. You need this glycerol backbone to reassemble the incoming fatty acids, and any made locally, into triglycerides for storage.
So even if fatty acids get in, without that glycerol piece from glucose, they can't be stored properly.
Correct. It's crucial for esterification in the process of attaching fatty acids to glycerol to make triglycerides. So insulin is necessary for both getting fatty acids into the cell and providing the means to store them and preventing their breakdown and release. It's promoting storage from multiple angles.
It really covers all the bases for fat storage. So what happens when insulin is deficient, particularly regarding fat metabolism? Things must go haywire.
Completely haywire. All those fat storing and breakdown inhibiting effects are reversed. This leads to a dramatic increase in fat mobilization and utilization for energy. It's extreme in severe diabetes.
How does that mobilization happen?
That hormone-sensitive lipase, the breakdown enzyme inside fat cells, becomes strongly activated because insulin isn't there to inhibit it. This causes rapid hydrolysis or breakdown of stored triglycerides.
Releasing fat back out.
Yes. Large quantities of fatty acids and glycerol pour out of the adipose tissue into the bloodstream. Plasma-free fatty acid concentrations shoot up. The source has a figure showing this happens almost immediately in experimental insulin deficiency. These free fatty acids then become the main energy source for most tissues, except the brain.
The body switches to burning fat heavily. What does this do to cholesterol levels?
This increased fat metabolism also impacts cholesterol and phospholipids. The excess fatty acids flooding the plasma cause the liver to convert some of them into phospholipids and cholesterol. These are then packaged into lipoproteins and released into the blood.
More lipoproteins in the blood.
Yeah. In severe insulin deficiency, plasma lipoprotein concentrations can triple. This results in very high plasma lipid levels, especially cholesterol. And this high lipid load strongly promotes the development of atherosclerosis in patients with chronic diabetes.
Leads to those vascular complications. And a really dangerous acute consequence is the development of ketosis and metabolic acidosis. How does that happen?
This occurs because when the liver is flooded with all those fatty acids in the absence of sufficient insulin, it breaks them down incredibly rapidly through beta oxidation. This process generates massive amounts of acetyl-CoA.
Acetyl-CoA.
Yeah.
The entry point to the energy cycle.
Usually, yes. But in the absence of insulin, peripheral tissues aren't efficiently using glucose or acetyl-CoA derived from fat, and the liver itself can't process all of the acetyl-CoA it's producing through its own energy cycle.
So there's a huge bottleneck.
A huge bottleneck. So a large portion of this excess acetyl-CoA gets diverted into another pathway. The liver converts it into acetoacetic acid. This acetoacetic acid is then released into the blood.
Acetoacetic acid, okay. And the body can't handle all of that.
No. The source explains that the absence of insulin also impairs the ability of peripheral tissues to use this acetoacetic acid efficiently. So it starts to accumulate rapidly in the body fluids.
Builds up in the blood.
Builds up, and some of it gets converted into two related compounds, beta-hydroxybutyric acid and acetone. Together, these three acetoacetic acid, beta-hydroxybutyric acid, and acetone are known as ketone bodies.
Ketone bodies, hence ketosis.
Exactly. When ketone bodies accumulate in large quantities, it's called ketosis. And because two of them, acetoacetic acid and beta-hydroxybutyric acid, are quite strong acids, their buildup causes a severe drop in the body's pH. That's metabolic acidosis.
A dangerous drop in pH.
Very dangerous. The source warns this can rapidly lead to diabetic coma and death, especially if the blood pH falls below about 7.0. The body tries to compensate, of course, increased breathing to blow off CO2, kidney actions, but in severe acute insulin deficiency, these compensations are often overwhelmed.
So without insulin, fat breakdown runs rampant, flooding the body with acidic byproducts. A critical situation. Let's shift to the third macronutrient protein. Insulin promotes synthesis and storage here too.
Yes, insulin promotes the formation and storage of proteins and prevents their degradation. This is especially important after a meal when amino acids from digested protein are available.
The mechanisms are less understood than for carbs and fat.
The source says they're less completely defined, but it lists several key actions. First, insulin stimulates the transport of many amino acids into cells. It mentions specific ones like valine, leucine, isoleucine, tyrosine, phenylalanine. It gets the building blocks inside where they're needed.
Helps import the necessary components.
Right. Second, it increases the rate at which ribosomes translate messenger RNA into new proteins. The source suggests insulin acts almost like an on switch for the ribosomes, boosting protein production.
Turns on the protein factories.
Third, over longer periods, insulin increases the rate of DNA transcription in the nucleus. This leads to more RNA being made and subsequently even more protein synthesis, particularly for metabolic enzymes involved in storage.
So more blueprints and faster production. What about breakdown?
Insulin also inhibits protein catabolism, the breakdown of existing proteins. It decreases the release of amino acids from tissues, especially muscle. The source suggests this might happen partly by diminishing the activity of lysosomes, the cell's recycling centers.
Protects existing protein.
And finally, by suppressing gluconeogenesis in the liver, which, remember, uses amino acids as fuel insulin, helps conserve the body's amino acids, keeping them incorporated in protein stores rather than being burned for energy or turned into glucose.
So overall, insulin strongly promotes protein building and prevents protein loss.
Correct. It's absolutely essential for maintaining the body's protein mass and structure.
Which logically means that insulin deficiency must cause protein depletion.
Exactly the opposite effect happens. Without sufficient insulin, protein storage basically halts, protein catabolism increases, protein synthesis stops.
So the balance shifts entirely to breakdown.
Entirely. This results in large quantities of amino acids being dumped into the plasma. These are then used for energy directly or get converted to glucose by the liver via that now unrestrained gluconeogenesis pathway. This whole process leads to increased urea excretion in the urine as the body gets rid of the nitrogen from the broken down amino acids.
And the physical effects.
This loss of body protein leads to wasting of tissues and extreme weakness or lack of energy, a condition called asthenia. This happens even if the individual is eating plenty of food. Polyphagia is common. In severe untreated diabetes, this protein wasting can become profound and contribute significantly to the overall deterioration.
A really devastating effect on body structure and function. The source also mentions an interesting interaction between insulin and growth hormone.
Yes, a synergistic effect on overall body growth. This is a key point.
Synergistic meaning they work better together.
Much better together than either alone. Because insulin is so critical for protein synthesis, the source states it's just as essential as growth hormone for promoting growth.
So growth hormone needs insulin's help.
It seems so. The source describes experiments in animals where both the pancreas, insulin source, and the pituitary gland, growth hormone source, were removed. Giving back either insulin alone or growth hormone alone resulted in almost no growth. But giving both hormones back together resulted in dramatic growth.
Wow. Clear synergy. Why do they work so well together?
The source speculates it might be because each hormone promotes the cellular uptake of different sets of amino acids, all of which are required together for synthesizing the full range of proteins needed for growth. They complement each other.
They need each other to supply all the necessary building blocks effectively. Okay, we've covered what insulin does and the impact of its absence.
Yeah.
Now let's dig into how the beta cell knows when to release insulin. What triggers secretion?
The primary, most important controller of insulin secretion is the blood glucose concentration itself.
Blood sugar levels drive it.
Fundamentally, yes. The source details the cellular mechanisms within the beta cell. Beta cells have a high density of a specific type of glucose transporter called GLUT2.
GLUT2, not GOUT4 like in muscle and fat.
Correct. GLUT2 allows glucose to enter the beta cell at a rate that's directly proportional to the glucose concentration in the blood, at least within the typical physiological range. So GLUT2 acts as the initial glucose sensor.
The cell effectively sees the external glucose level through these transporters.
Pretty much. Once inside the beta cell, the glucose is immediately phosphorylated. A phosphate group is added by an enzyme called glucokinase.
Glucokinase, is that important?
Extremely important. The source highlights glucokinase as being the rate-limiting step for glucose metabolism within the beta cell. And critically, it's considered the major mechanism by which the beta cell actually senses the blood glucose level and adjusts insulin secretion accordingly. It translates glucose concentration into an internal metabolic signal.
So glucokinase is the key sensor enzyme. How does that metabolic signal lead to insulin release?
The glucose 6-phosphate produced by glucokinase enters the cell's metabolic pathways and gets oxidized. This process generates ATP, the cell's energy currency.
More glucose means more ATP.
Exactly. And these increased ATP levels inside the beta cell have a specific target. They inhibit ATP-sensitive potassium channels in the cell membrane.
So high ATP closes potassium channels. What does that do?
Blocking these channels prevents positively charged potassium ions from leaving the cell. This causes a buildup of positive charge inside, leading the cell membrane to depolarize its electrical potential changes.
Depolarization. And that usually opens calcium channels in excitable cells.
Precisely the case here too. The depolarization opens voltage-gated calcium channels in the beta cell membrane. Calcium ions, which are more concentrated outside the cell, then flow into the cell.
Calcium rushes in, and calcium is often the trigger for secretion.
It is. This influx of calcium ions is the crucial signal that triggers the final step, the fusion of those docked insulin-containing secretory granules with the cell membrane. This fusion releases the granule contents insulin and C-peptide into the extracellular fluid via exocytosis.
Wow. It's a beautiful, precise sequence. Glucose entry via GLUT2, sensing and phosphorylation by glucokinase, ATP generation, potassium channel closure, depolarization, calcium influx, and finally, insulin secretion.
A really elegant example of stimulus secretion coupling, how a nutrient signal is transduced into a hormonal response. The source also notes that other nutrients, particularly certain amino acids, can also be metabolized by beta cells to generate ATP and stimulate insulin secretion, although glucose is the main driver.
What about other hormones or the nervous system influencing this process? Can they modulate it?
Yes, absolutely. They fine-tune the response. Hormones like glucagon itself and gut hormones like GLP-1 and GIP, plus the neurotransmitter acetylcholine, can enhance glucose-stimulated insulin secretion. They often work by increasing intracellular levels through other signaling pathways.
So they can boost the signal from glucose.
They can. But importantly, they generally don't cause major insulin release without the presence of glucose. They potentiate the glucose effect. Conversely, somatostatin from neighboring delta cells and norepinephrine from sympathetic nerves acting via alpha-adrenergic receptors actually inhibit insulin secretion, mainly by interfering with the exocytosis process.
So there are accelerators and brakes modifying the main glucose signal, and some diabetes medications target this specific pathway.
Yes, a major class of drugs for type 2 diabetes, the sulfonylureas, work precisely here. They bind to and block those ATP-sensitive potassium channels in the beta cell membrane,
Forcing them closed even if ATP isn't super high.
Exactly. This artificially closes the channels, causes depolarization, calcium influx, and stimulates insulin secretion. It helps lower blood glucose in patients whose beta cells are still functional, but perhaps not responding strongly enough to glucose alone.
Clever mechanism. Okay, let's look at the bigger picture of controlling insulin secretion. How does the body fine-tune this release moment by moment based on glucose levels?
As we said, blood glucose is the main driver. At normal fasting levels, around 80 to 90 mg 100 milliliters, insulin secretion is minimal, just a basal level. But if blood glucose suddenly increases, say after a meal, the beta cells respond with a characteristic two-stage pattern.
2 stages.
Yes. First, there's an immediate rapid surge of insulin release. It peaks within 3 to 5 minutes, maybe reaching 10 times the basal rate. This is thought to be the release of preformed insulin that's already docked and ready to go in granules right near the membrane.
An initial burst of readily available insulin. What's the second stage?
After that initial peak, secretion drops slightly for about 5 to 10 minutes. Then it begins to rise again more gradually, reaching a second higher plateau. This second phase starts around 15 minutes after the glucose stimulus and can last for hours if glucose remains high. It represents the release of other preformed insulin granules, plus the synthesis and release of newly formed insulin.
So a quick immediate response, followed by a more sustained release involving new production. And this system creates a powerful feedback loop with blood glucose.
A classic and crucial negative feedback loop. As blood glucose rises above the threshold, around 100 mg 100 milliliters, insulin secretion increases very steeply. The source mentions it can reach levels 10 to 25 times the basal rate at very high glucose concentrations, like 400 to 600 mg 100 milliliters.
A massive increase in output at high glucose.
Massive. But just as importantly, when blood glucose starts to fall back towards the normal range, insulin secretion shuts off almost immediately within 3 to 5 minutes.
Who's super responsive on the way down, too.
Yes. This tight feedback ensures that high glucose is quickly brought down by the high insulin response, but also that insulin levels don't stay inappropriately high once glucose normalizes, which could cause hypoglycemia. It's all about maintaining that balance.
Besides glucose, what other factors are significant stimulators of insulin release? You mentioned amino acids.
Yes, certain amino acids, particularly arginine and lysine, are potent stimulators. Interestingly, while they cause only a modest increase in insulin secretion on their own, they significantly potentiate the effect of glucose.
Potentiate, they make the glucose effect stronger.
Exactly. If you have high glucose and high amino acids together, the insulin secretion can be nearly double what high glucose alone would cause. This is important after a protein-containing meal, ensuring the body can properly utilize those incoming amino acids for protein synthesis, driven by the boosted insulin levels.
Makes sense. And the gut hormones you mentioned earlier, the incretins.
Right. Hormones released from the gastrointestinal tract in response to food, like gastrin, secretin, cholecystokinin, and especially GLP-1, glucagon-like peptide 1, and GIP, glucose-dependent insulinotropic polypeptide. These last two, GLP-1 and GIP, are known as the incretins.
Incretins, what do they do?
They cause a moderate increase in insulin secretion. What's really interesting is they are released after you eat, but before glucose and amino acids are fully absorbed into the bloodstream. So they cause an anticipatory rise in insulin.
Getting the system ready for the incoming nutrients.
Precisely. They prime the pancreas. And like amino acids, they also increase the sensitivity of the beta cells to the glucose stimulus itself, again, potentially doubling the insulin response at a given glucose level. This incretin effect is significant enough that therapies mimicking or enhancing incretin action are now important treatments for type 2 diabetes.
Clever approach, leveraging the body's own anticipatory signals. What about other hormones in the nervous system? Do they play a role in modulating insulin secretion?
They do. Several other hormones, including glucagon itself, growth hormone, cortisol, and even progesterone and estrogen, can increase insulin secretion or potentiate the glucose stimulus.
So things like stress hormones can actually boost insulin.
Acutely, yes, or make the glucose effect stronger. However, the source cautions that prolonged high secretion of some of these, like growth hormone or cortisol, as seen in conditions like acromegaly or Cushing syndrome, can put chronic strain on the beta cells. This can potentially lead to beta cell exhaustion and the development of diabetes.
A potential downside to long-term overstimulation and the nervous system.
The pancreatic islets are richly innervated by both branches of the autonomic nervous system. Parasympathetic stimulation, often associated with digestion and nutrient absorption, tends to increase insulin secretion, especially when glucose is already elevated.
The rest and digest system boosts insulin.
Right. Conversely, sympathetic stimulation, associated with the fight or flight response or stress like hypoglycemia, tends to decrease insulin secretion and increase glucagon secretion. This makes sense. During low blood sugar, you want to stop insulin from lowering glucose further and get glucagon to raise it.
So the nervous system integrates insulin control with the body's overall state.
Exactly. And this nervous control likely integrates signals from glucose-sensing neurons in the brainstem and hypothalamus, as well as signals from the periphery. It's a complex, multifactorially controlled process, all aimed at dynamic regulation based on nutrient availability and the body's needs.
Okay. The source summarizes all this by talking about how insulin, along with other hormones, facilitates the body's ability to switch between using carbohydrates and fats for energy.
Yes, this metabolic switching is fundamental to energy homeostasis. As we've discussed, insulin strongly promotes carbohydrate utilization and storage while actively suppressing fat use.
And the absence of insulin does the opposite.
The absence or low levels of insulin promote fat utilization for energy, again, except in the brain. The principal signal dictating which fuel source is predominantly used is the blood glucose concentration itself, acting via its control over insulin secretion.
So high glucose, high insulin favors carb use. Low glucose, low insulin echo favors fat use. That's the core switch.
That's the core switch. But other hormones contribute to this switching, especially over different timescales or under specific conditions.
Like growth hormone and cortisol.
Right. They tend to respond to more prolonged periods of hypoglycemia, maybe over hours or days. Their effect is to inhibit glucose utilization by most cells and promote fat utilization. But these effects develop much more slowly than the rapid insulin-glucagon switch.
Slower, longer-term adjustments. What about epinephrine? Adrenaline?
Epinephrine is important during acute stress, like heavy exercise, circulatory shock, or even anxiety. It acts rapidly. It increases plasma glucose levels, mainly by stimulating glycogen breakdown in the liver, and it strongly increases plasma fatty acid levels by activating lipase in adipose tissue.
So it mobilizes both fuel types.
It mobilizes both, but quantitatively, its effect on increasing fatty acids is often greater. So, especially during stress situations, epinephrine powerfully shifts metabolism towards fat utilization.
These other hormones provide slower adjustments or handle acute stress responses, complementing the main insulin-glucagon system. Now, let's formally introduce glucagon, the main counterpoint to insulin.
Right. Glucagon. Secreted by the alpha cells in the islets of Langerhans, primarily when blood glucose levels fall. Its actions are, for the most part, diametrically opposed to insulin's.
And its main job?
Its most critical function is to increase blood glucose concentration when it drops too low. It prevents hypoglycemia.
What is it chemically?
Yeah.
And how potent is it?
Well, glucagon is also a polypeptide, but smaller than insulin. It's a single chain of 29 amino acids with a molecular weight of about 3/34 of an 85. And it's very potent at raising blood glucose. It's often called the hyperglycemic hormone for a good reason.
How potent?
The source gives an example. Just 1 microgram per kilogram of body weight injected can raise blood glucose by about 20 ml of 100 milliliters. That's a 25% increase within only 20 minutes. It works fast and effectively to mobilize glucose.
Very powerful. What are its main mechanisms for achieving this? How does it raise blood glucose?
Glucagon primarily acts on the liver. It has 2 major effects there. 1, it causes the breakdown of stored liver glycogen, a process called glycogenolysis. 2, it increases the synthesis of new glucose in the liver from other sources, which is gluconeogenesis.
Breakdown of stored glucose and making new glucose. Both boost output.
Exactly. Both actions enhance the amount of glucose the liver releases into the bloodstream.
Let's look at the glycogen breakdown first. How does glucagon trigger that rapid release from liver stores? It's a famous pathway, isn't it?
It is. It's achieved through a very well-studied second messenger system involving cyclic AMP or CMP. It's one of the classic examples.
How does the cascade work?
Okay, when glucagon binds to its specific receptor on a liver cell membrane, it activates an enzyme called adenyl cyclase. This enzyme then converts ATP inside the cell into cyclic AMP, CAMP.
CAMP is the second messenger.
Right. This rise in CAMP then activates another enzyme, protein kinase A. Protein kinase A then phosphorylates and activates another kinase called phosphorylase kinase. And phosphorylase kinase, in turn, activates the enzyme that actually breaks down glycogen phosphorylase, specifically converting it from the less active B form to the highly active A form.
Wow, a cascade of kinase and activating kinases.
It is. And the activated glycogen phosphorylase A then starts rapidly breaking down glycogen into glucose 1-phosphate units. These are then converted to glucose 6-phosphate, and finally, the phosphate is removed by glucose phosphatase, allowing free glucose to be released from the liver cell.
What's the significance of this multi-step cascade? Why not just have glucagon directly activate the breakdown enzyme?
The source highlights two key things about this cascade. First, as you said, it's one of the most thoroughly studied SAMP-mediated pathways, so it's fundamentally important in cell biology. Second, and crucially for physiology, it acts as a powerful amplifying mechanism.
Amplifying?
Yes. At each step in the cascade, one activated enzyme molecule can activate many molecules of the next enzyme in the chain. So a single glucagon molecule binding to a receptor can lead to the activation of millions of glycogen phosphorylase molecules. This massive amplification explains how tiny amounts of glucagon circulating in the blood can cause such a large and rapid increase in glucose release from the liver.
A tiny signal gets massively boosted to unlock a huge amount of stored energy very quickly. That's clever. What about its effect on making new glucose, gluconeogenesis?
Glucagon also stimulates gluconeogenesis in the liver. And importantly, this effect continues even after the liver's glycogen stores have been depleted, say during prolonged fasting.
How does it boost gluconeogenesis?
It increases the rate at which the liver takes up amino acids from the blood, which are major precursors for gluconeogenesis. And it activates key enzymes within the gluconeogenesis pathway itself, promoting the conversion of these amino acids and other precursors like lactate or glycerol into glucose. It particularly activates enzymes involved in bypassing irreversible steps in glycolysis, like the conversion of pyruvate to phosphenylpyruvate, which is often a rate-limiting step.
So it can mobilize stored glucose rapidly and stimulate the sustained production of new glucose from other sources. Does glucagon have significant effects beyond glucose metabolism?
The source mentions other effects, but notes they generally occur only at concentrations higher than those typically found circulating in the blood under normal physiological conditions.
So probably less important day to day.
Likely less important than its glucose effects, yes. But at these high concentrations, one important effect is that glucagon can activate hormone-sensitive lipase in adipose cells.
Like insulin deficiency does.
Yes, leading to increased release of fatty acids into the blood, making them available for energy. It also inhibits triglyceride storage in the liver itself, which might further increase the availability of fatty acids for other tissues.
Okay. Any other minor effects?
At very high concentrations, the source mentions it can enhance the strength of heart contraction, increase blood flow through the kidneys, enhance bile secretion, and inhibit gastric acid secretion. But again, these are likely less physiologically relevant under normal circumstances compared to its profound impact on glucose.
So glucagon is primarily the glucose mobilizer, the anti-hypoglycemia hormone. How is its secretion regulated? What tells the alpha cells to release it?
Just like insulin, blood glucose concentration is the most potent factor controlling glucagon secretion. But the effect is precisely the opposite to its effect on insulin.
High glucose inhibits glucagon.
Exactly. Increased blood glucose strongly inhibits glucagon secretion. The source shows data illustrating that as blood glucose rises above normal fasting levels, around 90 millijoule 100 milliliters, glucagon secretion decreases. Conversely, when blood glucose falls below this level into the hypoglycemic range, glucagon secretion increases sharply, potentially rising several-fold.
So low glucose is the major stimulus for glucagon release.
That's its key role. It's secreted in large amounts during hypoglycemia, specifically to stimulate liver glucose output and raise blood glucose levels back towards normal, directly counteracting any residual insulin effect and the glucose drop itself.
Okay, low glucose stimulates glucagon. What about amino acids? They stimulated insulin. What do they do to glucagon?
This is interesting. High concentrations of certain amino acids in the blood, especially alanine and arginine, also stimulate glucagon secretion.
They stimulate both insulin and glucagon. That seems weird.
It does seem counterintuitive at first, since their effects on glucose are opposite. But the source offers a rationale. The glucagon secreted in response to a protein-rich meal promotes the rapid conversion of those absorbed amino acids into glucose by the liver via gluconeogenesis.
So the amino acids stimulate insulin to help store them as protein, But they also stimulate glucagon to turn some of them quickly into glucose.
Exactly. This helps prevent hypoglycemia that might otherwise occur if insulin were stimulated strongly by amino acids without a simultaneous carbohydrate load. It ensures adequate glucose availability.
A clever balance after a protein meal. What about exercise? Does that affect glucagon?
Yes. Blood glucagon levels often increase significantly, maybe fourfold to fivefold, during exhaustive exercise.
Why would that happen?
This increase is thought to be important for preventing blood glucose from falling too low during prolonged or intense activity by stimulating liver glucose output to match the increased muscle glucose uptake. The exact cause of the glucagon rise during exercise isn't fully understood, but the source suggests contributing factors might include stimulation by circulating amino acids released from muscle, or perhaps beta-adrenergic stimulation of the alpha cells via the sympathetic nervous system.
So glucagon release is primarily driven by low glucose, ensuring energy availability when needed, but also influenced by amino acids and exercise. Let's briefly touch on the third hormone mentioned from the islets, somatostatin, from the delta cells.
Right, somatostatin. Secreted by the delta cells, it's a relatively small peptide, 14 amino acids, with a very short half-life in the blood, only about 3 minutes.
Quick acting, quickly gone. What stimulates its release?
Its secretion seems to be stimulated by almost all factors related to food ingestion. So increased blood glucose, increased amino acids, increased fatty acids, and also various gastrointestinal hormones released after a meal.
So food intake triggers somatostatin, and its main action is inhibitory.
Yes, generally inhibitory. Locally, right within the islets of Langerhans, somatostatin acts in a paracrine manner to depress the secretion of both insulin and glucagon from the neighboring beta and alpha cells.
A local break on both the others. Does it have effects elsewhere?
It does. Somatostatin released into the circulation also has widespread inhibitory effects in the gastrointestinal tract. It decreases motility of the stomach, duodenum, and gallbladder, and it also decreases both secretion and absorption throughout the GI tract.
Slows down the whole digestive and absorptive process. What's the suggested overall role for pancreatic somatostatin?
The source suggests its principal role might be to extend the period over which the nutrients absorbed from a meal are assimilated into the blood. By slowing down GI processing and also by depressing both insulin and glucagon secretion, which would normally accelerate nutrient utilization and storage, somatostatin might effectively spread out the availability of nutrients to the tissues over a longer time period, like a modulator smoothing out the post-meal nutrient surge.
Interesting. Like a pause button or slow motion control on the whole post-meal metabolic response. And the source notes it's the same chemical as a hypothalamic hormone.
Yes, pancreatic somatostatin is chemically identical to the growth hormone inhibitory hormone, GHIH, secreted by the hypothalamus, which acts on the anterior pituitary gland to suppress the secretion of growth hormone. Same molecule, different inhibitory roles in different parts of the endocrine system.
Fascinating. Okay, let's try to pull all this together now into a summary of how blood glucose is normally regulated so tightly.
Right. As we've said, the body maintains remarkably tight control over blood glucose concentration. Normal fasting levels are typically in the range of 80 to 90 milliliters, 100 milliliters. After a carbohydrate-containing meal, levels might rise to under 2140, but usually return back to the baseline within about 2 hours. Even during prolonged fasting or starvation, gluconeogenesis usually manages to maintain that fasting level.
How is this incredibly high degree of control achieved? What are the key mechanisms the source summarizes?
It lists several crucial overlapping mechanisms. First, the liver acts as a major blood glucose buffer system.
The glycogen storage and release we discussed.
Exactly. By taking up and storing glucose as glycogen when levels are high after a meal, absorbing maybe 2/3 of the glucose load, and then releasing glucose back into the blood between meals, the liver significantly dampens blood glucose fluctuations. The source estimates it reduces fluctuations by about 2/3 compared to what they would be without this buffering capacity. Again, noting severe liver disease impairs this significantly.
Okay, liver buffering is number one. What's next?
Second, the feedback control systems of insulin and glucagon are paramount. They operate like a push-pull system. When glucose goes up, insulin secretion rises sharply to push it back down by promoting uptake, utilization, and storage. When glucose goes down, insulin secretion falls, and glucagon secretion rises to push glucose back up by stimulating liver output.
The two main hormonal actors keeping levels stable. Which one is more important?
The source suggests that under normal conditions, the insulin feedback mechanism is probably more important for minute-to-minute regulation. But the glucagon mechanism becomes critically important in preventing severe hypoglycemia, especially during starvation or prolonged exercise.
Okay, liver buffer, insulin glucagon feedback. What else?
Third, in situations of severe hypoglycemia, there's a direct effect of low glucose on the hypothalamus in the brain. This triggers activation of the sympathetic nervous system.
Fight or flight response kicks in.
Yes, this leads to the release of epinephrine, adrenaline, from the adrenal medulla. Epinephrine acts rapidly on the liver to cause glycogenolysis and glucose release, providing a crucial emergency backup system to counteract dangerously low glucose levels.
An emergency glucose boost. Is there anything else?
Fourth, over longer periods of time, hours to days, if hypoglycemia persists, other hormones play a role. Growth hormone and cortisol secretion increases.
The slower acting ones we mentioned.
Right. These hormones act more slowly to decrease the rate of glucose utilization by most peripheral tissues, essentially making them more reliant on fat. This glucose-sparing effect helps to gradually restore blood glucose levels back towards normal during prolonged fasting or other chronic hypoglycemic stresses.
So, multiple layers of control. Rapid buffering, fast hormonal feedback, emergency sympathetic response, and slower hormonal adjustments. Given that most tissues can use fats and proteins for energy, why is it so critically important to maintain glucose within this narrow range? Why all this effort?
The source is very clear on this. Glucose is essentially the only nutrient that can be used optimally for energy by the brain, the retina, and the eye, and the germinal epithelium of the gonads under normal conditions.
The brain is the main reason.
The brain is the main reason. It's highly dependent on a continuous supply of glucose from the blood. That's why most of the glucose produced by the liver via gluconeogenesis during the interdigestive periods between meals or fasting is specifically destined for the brain.
And why is low insulin between meals important for the brain?
It's crucial. If insulin levels remained high during fasting, the peripheral tissues, especially muscles, would continue to take up large amounts of the limited glucose available. This could potentially deplete the blood glucose to levels that are too low to adequately supply the brain, leading to the hypoglycemic shock symptoms we discussed. Low insulin between meals ensures that glucose uptake by muscles and fat is minimized, sparing the glucose for the brain.
It prioritizes the brain's fuel supply. Okay, so low glucose is bad, mainly for the brain. What are the specific harms of having blood glucose levels that are too high for prolonged periods?
Chronic hyperglycemia, as seen in poorly controlled diabetes, causes several types of damage. First, high glucose concentrations in the extracellular fluid increase its osmotic pressure. This can draw water out of cells, leading to cellular dehydration.
Dehydration at the cellular level.
Yes. Second, when blood glucose exceeds the kidney's capacity to reabsorb it from the filtered fluid, a threshold usually around 180-200 millig, 100 milliliters, glucose starts spilling into the urine. This is called glycosuria.
Losing sugar in the urine.
Right. And this loss of glucose in the urine itself causes problems. The glucose molecules in the renal tubules exert an osmotic effect, preventing water from being reabsorbed properly. This leads to excessive water loss in the urine, a condition called osmotic diuresis.
Making you urinate a lot.
Exactly. Massive urine production, polyuria. This, in turn, causes overall body dehydration, both extracellular and intracellular, which then triggers intense thirst, polydipsia. These polyuria, dehydration, polydipsia are classic symptoms of uncontrolled diabetes.
And the long-term damage from high glucose.
This is perhaps the most serious consequence. Chronic high glucose levels cause damage to tissues throughout the body, especially blood vessels. This vascular damage leads to abnormal blood vessel function and structure, often resulting in inadequate blood supply to tissues over time.
Leading to major complications.
Yes. This significantly increases the risk for heart attack, stroke, end-stage kidney disease requiring dialysis or transplant, retinopathy, damage to the blood vessels in the eye, potentially causing blindness, and peripheral vascular disease leading to limb ischemia and potentially gangrene and amputation.
Devastating vascular consequences.
Yeah.
Any other tissues affected?
Yes. Chronic hyperglycemia also damages nerves, leading to peripheral neuropathy, causing pain, numbness, or weakness, often in the hands and feet, and dysfunction of the autonomic nervous system, affecting things like cardiovascular reflexes, bladder control, digestion, and sexual function. The exact mechanisms of how high glucose causes all this tissue damage are complex and still not fully understood, but likely involve glucose directly affecting cellular proteins and functions, often amplified by associated conditions like hypertension and atherosclerosis.
So high glucose is directly toxic to many tissues over the long term. This brings us squarely to diabetes mellitus, the condition where this intricate regulation goes wrong. Define it for us again based on the source.
Diabetes mellitus is defined as a syndrome characterized by impaired metabolism of carbohydrates, fats, and proteins. It's caused by either a lack of insulin secretion from the pancreas or a decreased sensitivity of the body's tissues to the effects of insulin, what we call insulin resistance.
Lack of insulin or resistance to insulin. What's the common metabolic outcome in both types?
In both type 1 and type 2 diabetes, the fundamental metabolic problem is that glucose cannot be efficiently taken up and utilized by most body cells, again except the brain, because of either the absence of insulin or the impaired response to it. This inevitably leads to elevated blood glucose levels, hyperglycemia, decreased glucose use by cells, and a compensatory unhealthy increase in the utilization of fats and proteins for energy.
Okay, let's differentiate the two main types. Type 1 diabetes first. What's the cause?
Type 1 diabetes is caused by destruction of the pancreatic beta cells, leading to an absolute deficiency of insulin secretion. The source mentions the destruction is often due to autoimmune processes, where the body's own immune system attacks the beta cells, possibly triggered by viral infections or other environmental factors in genetically susceptible individuals. Less commonly, it might be due to other injuries or degeneration of the islets.
On a typical onset.
Onset is usually in childhood or adolescence, the average age in the U.S. being around 14 years. That's why it used to be called juvenile diabetes. However, it can occur at any age. The onset is typically quite abrupt, developing rapidly over a period of days or weeks.
And the key consequences resulting directly from this lack of insulin.
The source highlights 3 major sequelae. 1. Markedly increased blood glucose levels. 2. Greatly increased utilization of fats for energy, leading to metabolic acidosis and high blood cholesterol. And 3. Depletion of the body's protein stores. These stem directly from the loss of insulin's metabolic actions we've discussed.
How common is type 1?
It accounts for a smaller proportion of all diabetes cases, roughly 5% to 10%.
Okay. Now, type 2 diabetes, which is much more prevalent. What's the underlying cause here?
The fundamental defect in type 2 diabetes, at least initially, is decreased sensitivity of the target tissues, especially muscle, liver, and fat, to the metabolic effects of insulin. This is known as insulin resistance.
Resistance to insulin signal. And the onset.
Type 2 typically develops much more gradually than type 1, often over many years. It usually occurs in adults, typically after age 30, with the average age of onset often being between 50 and 60. Hence, it was previously called adult onset diabetes. However, alarmingly, due to rising rates of obesity, it's now being diagnosed increasingly frequently in younger individuals, including adolescents and children.
And its prevalence.
Type 2 diabetes accounts for the vast majority of cases, about 90% to 95%.
So the core issue is insulin resistance. The tissues don't respond properly.
Exactly. And the source notes an important point. In the early stages of type 2 diabetes, plasma insulin levels are often not low. In fact, they are frequently elevated. This condition is called hyperinsulinemia.
Why would insulin be high if the problem is resistance?
It's a compensatory response. The insulin resistance impairs glucose utilization and storage by the tissues. This causes blood glucose levels to rise. The elevated glucose then stimulates the pancreatic beta cells, which are still functional initially, to secrete even more insulin in an attempt to overcome the resistance and maintain normal blood glucose levels.
So the pancreas is working overtime to try and make up for the lack of response.
Precisely. It's trying to shout louder, metabolically speaking, to get the message through. But eventually, this compensation may fail.
What drives the development of this insulin resistance?
The development of insulin resistance is a gradual process, and the most significant risk factor by far is excess weight gain and obesity, particularly an accumulation of abdominal or visceral fat. While the exact mechanisms are complex, the source suggests that although a decrease in the number of insulin receptors on cells might play a small role, the primary defects are likely within the intracellular signaling pathways after insulin binds to its receptor.
Problems inside the cell's response mechanism.
Yes. There's growing evidence, mentioned in the source, that the accumulation of lipids, fats, within tissues like muscle and liver, often associated with obesity, can directly interfere with these insulin signaling pathways, leading to resistance.
And this insulin resistance is often part of a larger cluster of issues called the metabolic syndrome.
Yes, the source describes insulin resistance as being a central component of what's often called the metabolic syndrome, or sometimes syndrome X. This syndrome is characterized by a cluster of risk factors occurring together.
What are the features of metabolic syndrome?
Typically, it includes obesity, especially excess abdominal fat, insulin resistance itself, fasting hyperglycemia, elevated fasting blood glucose, though maybe not yet diabetic levels, lipid abnormalities, specifically high blood triglycerides and low levels of HDL, the good cholesterol, and hypertension, high blood pressure.
Constellation of risk factors.
Exactly. And the source strongly links these features, particularly insulin resistance, to the accumulation of excess fat, especially visceral fat around the internal organs. Insulin resistance is considered the primary underlying defect leading to the high blood glucose seen in this syndrome. And the major adverse consequence of metabolic syndrome is a greatly increased risk of cardiovascular disease, atherosclerosis, heart attacks, strokes.
So metabolic syndrome significantly raises cardiovascular risk.
Hugely. The metabolic abnormalities themselves increase risk. The insulin resistance strongly predisposes individuals to eventually developing full-blown type 2 diabetes, and type 2 diabetes itself is a major cause of cardiovascular disease. It's a dangerous combination.
Are there other causes of insulin resistance or type 2 diabetes besides the common pathway involving obesity and metabolic syndrome?
Yes, although obesity is the most common driver. The source lists several other conditions, both acquired and genetic, that can cause or contribute to insulin resistance by impairing insulin signaling. It provides a table with examples.
Like what?
Examples include polycystic ovary syndrome, PCOS, which often involves insulin resistance and hyperinsulinemia along with hormonal changes. Excess glucocorticoids, as seen in Cushing's syndrome, can cause insulin resistance. Excess growth hormone and acromegaly can do the same. Even pregnancy can induce a state of insulin resistance, sometimes leading to gestational diabetes. Other rarer causes include certain genetic forms of obesity, lipodystrophy, abnormal fat distribution, the presence of autoantibodies against the insulin receptor, or mutations in the insulin receptor gene, or other genes involved in insulin action like PPARO, and conditions like hemochromatosis, iron overload.
So a variety of factors can disrupt that critical insulin signaling pathway and lead to resistance. How does type 2 diabetes typically progress if it's not managed effectively? Does the resistance just get worse?
It often starts with that compensated insulin resistance, where high insulin levels manage to keep blood glucose relatively normal, maybe with only moderate hyperglycemia after meals. But over time, especially if the resistance persists or worsens, the pancreatic beta cells can begin to fail.
They get exhausted.
That's one theory mentioned, beta cell exhaustion from the chronic overstimulation. Another possibility suggested is that the beta cells themselves can be damaged, perhaps by the same factors causing peripheral resistance, like lipid accumulation, lipotoxicity within the islets. Whatever the exact mechanism, the beta cells eventually become unable to sustain the high level of insulin secretion needed to overcome the resistance.
So insulin production starts to fall.
Yes, relative to the level needed. When this happens, insulin secretion becomes insufficient to control blood glucose effectively, leading to more severe and persistent hyperglycemia, especially after meals, and eventually even in the fasting state. The source notes that some obese individuals never develop overt diabetes because their pancreas manages to keep up the high insulin output indefinitely, while others do progress to diabetes as their pancreatic function declines. Genetic factors likely play a significant role in determining this beta cell resilience or susceptibility to failure.
A combination of resistance and eventual beta cell dysfunction. How is type 2 diabetes treated then?
Treatment strategies vary depending on the stage and severity. In many cases, especially early on, lifestyle modifications are the first line and can be very effective. This includes increasing physical activity, restricting calorie intake, and achieving weight loss. These measures can significantly improve insulin sensitivity and glucose control.
What about medications?
If lifestyle changes aren't sufficient, various medications are available. Some drugs work primarily by increasing insulin sensitivity in peripheral tissues, like thiazolidinediones. Others work mainly by suppressing excessive glucose production by the liver, like metformin, often a first-line drug. Still others act by stimulating the pancreas to release more insulin, like the sulfonylureas we discussed earlier. Often combinations of drugs with different mechanisms are used.
And if those stop working?
In the later stages of type 2 diabetes, if beta cell function declines significantly, the patient may no longer produce enough insulin, even with stimulation. At that point, insulin injections become necessary to control blood glucose, similar to the treatment for type 1 diabetes.
The source mentions some newer drug approaches as well, targeting different pathways.
Yes, several newer classes have become important. One group targets the incretin system we talked about. There are drugs that mimic the action of GLP-1 receptor agonists, enhancing glucose-dependent insulin secretion and having other beneficial effects. Another related class inhibits the enzyme DPP-4, which normally breaks down the body's own incretins, GLP-1 and GIP. By blocking DPP-4, these inhibitors prolong the action of natural incretins, increasing insulin secretion.
So boosting that natural gut-pancreas signaling.
Yeah.
What about the SGLT2 inhibitors? They work differently, right, in the kidneys?
Yes, that's a completely different mechanism.
Yeah.
SGLT2 inhibitors target the sodium-glucose cotransporter 2, which is located in the proximal tubules of the kidneys.
What does SGLT2 normally do?
Under normal conditions, SGLT2 is responsible for reabsorbing about 90% of the glucose that gets filtered out of the blood by the kidneys, returning it back into the circulation.
So it saves glucose from being lost in urine.
Exactly. The SGLT2 inhibitors, drugs often ending in glyphosate, block this transporter. By blocking SGLT2, they prevent the kidneys from reabsorbing much of the filtered glucose.
So the glucose stays in the filtrate and gets excreted.
Precisely. This results in a large amount of glucose being excreted in the urine, sometimes dozens of grams per day. This loss of glucose from the body effectively lowers blood glucose levels, independent of insulin action.
That's a novel approach. Any other effects?
Yes. The source highlights that these SGLT2 inhibitors have shown significant benefits beyond just lowering glucose. Clinical trials have demonstrated they can protect against cardiovascular events, like heart failure, and slow the progression of kidney disease in patients with diabetes. The mechanisms for these protective effects are still being fully elucidated, but are thought to involve factors beyond glucose lowering, perhaps related to effects on blood pressure, kidney hemodynamics, and metabolism. They also cause that osmotic diuresis effect due to the glucose in the urine, which can lead to a modest reduction in blood pressure, but also carries a risk of dehydration or hypotension, especially if used with other diuretics or blood pressure medications.
So multiple benefits, but also potential side effects to watch for. Finally, the source mentions surgical options for type 2 diabetes.
Yes, for individuals who have severe obesity along with type 2 diabetes, bariatric surgery procedures like gastric bypass or vertical sleeve gastrectomy have emerged as highly effective treatment options.
How do they help with diabetes?
These surgeries lead to significant weight loss, which itself improves insulin sensitivity. But remarkably, they often result in dramatic improvement or even complete remission of type 2 diabetes, sometimes very rapidly after the surgery, even before substantial weight loss has occurred. This has led to the term metabolic surgery being used.
Remission of diabetes.
It's huge.
Why does it happen so quickly?
The rapid improvement suggests that the benefits go beyond just weight loss. The exact mechanisms are still under intense investigation, but are thought to involve complex changes in the secretion of gut hormones, like incretins, alterations in bile acid metabolism, changes in the gut microbiota, and possibly other factors related to the rerouting of the gastrointestinal tract. It's a fascinating area showing the powerful interplay between the gut and metabolic regulation.
A very complex picture for type 2 diabetes, involving resistance, potential beta cell failure, and a growing array of treatment options targeting different aspects of the disease. How do clinicians actually diagnose diabetes? What tests are used?
Diagnosis relies on detecting abnormal glucose metabolism, primarily through chemical tests on urine and blood. The source mentions a table comparing typical features of type 1 versus type 2, like age of onset, body mass, plasma insulin levels, etc., which can give clues.
What about specific tests? Urine glucose.
Simple tests can detect glucose in the urine, glycosuria. Normally, there's virtually no detectable glucose in urine. In diabetes, especially when blood glucose is high, glucose spills over into the urine and can be measured. The amount roughly correlates with severity and recent carbohydrate intake.
But blood tests are more definitive.
Yes. Fasting blood glucose is a cornerstone. Normal fasting glucose is typically 80-90 milligrammes 100 milliliters. The upper limit of normal is generally considered around 100 or maybe 110 milligrammes 100 milliliters. Levels consistently above 126 milligrammes 100 milliliters on fasting tests usually indicate diabetes. Levels between 100 and 125 might indicate prediabetes or impaired fasting glucose.
What about fasting insulin levels?
Measuring fasting insulin can sometimes help distinguish types. In type 1, fasting insulin is typically very low or undetectable. In early type 2, as we discussed, fasting insulin may be normal or even high due to compensatory hyperinsulinemia. However, insulin levels can be variable, so it's not always definitive alone.
Then there's the HbA1c test. What does that measure again?
Glycated hemoglobin, or hemoglobin A1c, HbA1c. This measures the percentage of hemoglobin molecules in red blood cells that have glucose attached to them. This attachment happens gradually and non-enzymatically over the lifespan of the red blood cell, which is about 120 days.
So it reflects average glucose over time.
Exactly. The higher the average blood glucose has been over the preceding 2 to 3 months, the higher the HbA1c level will be. It gives a much better picture of long-term glycemic control than a single blood glucose measurement, which can fluctuate significantly day to day. HbA1c is now widely used both for diagnosing diabetes, a level of 6.5% or higher is diagnostic, and for monitoring how well diabetes is being managed over time.
A very valuable long-term marker. What about the glucose tolerance test, GTT? Is that still used?
Yes, the oral glucose tolerance test, OGTT, is still considered a standard test, especially in borderline cases or for diagnosing gestational diabetes. It involves measuring fasting blood glucose, then having the person drink a standard amount of glucose solution, usually 75 grams, and then measuring blood glucose levels again periodically over the next 2 hours.
How does the response differ in diabetes?
In a person without diabetes, fasting glucose is normal. It rises after the glucose load, but usually stays below 100 milliliters at 2 hours and returns to baseline relatively quickly. In someone with diabetes, the fasting glucose may already be elevated, for example, 126. After the glucose load, the level rises much higher, typically exceeding 100 milliliters at the 2-hour mark, and the return to baseline is significantly delayed, often taking 4 to 6 hours or longer.
So it shows impaired ability to handle a glucose load.
Precisely. It indicates either insufficient insulin secretion or decreased insulin sensitivity or both. The source notes that measuring insulin levels during the OGTT can sometimes help differentiate. Type 1 typically shows a very blunted or absent insulin response, while type 2 often shows an elevated but still inadequate insulin response relative to the very high glucose levels.
Okay. And one last diagnostic clue mentioned is acetone breath.
Yes. In severe uncontrolled diabetes, particularly type 1 resulting in significant ketoacidosis, one of the ketone bodies, acetoacetic acid, can be spontaneously converted into acetone. Acetone is volatile and can be expelled in the breath, giving it a characteristic fruity or nail polish remover odor. Detecting acetone breath is a clinical sign suggesting the presence of significant ketosis. Measuring ketone bodies in the urine or blood can quantify the severity.
A direct sign of that dangerous metabolic derangement. Finally, the source connects the goals of treatment back to preventing those serious long-term complications we discussed earlier.
Right, because individuals with diabetes, especially type 2, are at such high risk for atherosclerosis, heart disease, chronic kidney disease, retinopathy, neuropathy, etc., often driven by the combination of hyperglycemia, abnormal lipid levels, high triglycerides, low HDL, often high LDL, and hypertension. The goals of treatment extend beyond just controlling blood sugar.
It's about managing the whole metabolic picture.
Exactly. The source notes that historically, treatment sometimes focused heavily on severe carbohydrate restriction. While this could help control blood glucose and reduce glucose loss in urine, it often didn't adequately address the underlying abnormalities in fat metabolism and might have even exacerbated them, potentially not reducing the risk of cardiovascular complications.
So the thinking has changed.
Yes. The current approach generally emphasizes achieving near-normal blood glucose control using appropriate medications, including insulin when necessary, while allowing for a more balanced diet that includes a reasonable amount of carbohydrates. The idea is that providing enough insulin or enhancing sensitivity to properly metabolize these carbohydrates helps to normalize fat metabolism as well, reducing the excessive fat breakdown and potentially lowering blood cholesterol and triglyceride levels.
Aiming for overall metabolic balance.
Right. And very often physicians will also prescribe medications specifically targeting lipid abnormalities, like statins to lower LDL cholesterol, and hypertension in patients with diabetes, as managing these associated risk factors is crucial for reducing the long-term risk of cardiovascular and other complications. Good glycemic control and good control of lipids and blood pressure are all key.
A comprehensive strategy targeting multiple risk factors to prevent those devastating long-term outcomes. Before we wrap up this deep dive, the source briefly mentions a condition called insulinoma, which is essentially the opposite problem, too much insulin.
Yes, insulinoma. It's a rare condition, usually caused by an adenoma, a benign tumor of the pancreatic beta cells. Sometimes, about 10-15% of cases, these tumors can be malignant and metastasize. The key feature is that the tumor autonomously secretes excessive amounts of insulin, regardless of blood glucose levels.
So the feedback control is lost.
completely lost. This leads to inappropriately high insulin levels, which can drive blood glucose dangerously low, causing hypoglycemia. The source mentions that in extreme cases, patients might need to consume huge amounts of glucose, over 1,000 grams per day, just to counteract the effects of the excess insulin and prevent severe hypoglycemia.
Wow. And this excessive insulin leads to insulin shock and hypoglycemia.
Exactly. The high insulin drives glucose out of the blood and into tissues, except the brain, and strongly suppresses liver glucose production. The resulting low blood glucose, hypoglycemia, primarily affects the central nervous system because, as we've stressed, the brain relies so heavily on glucose for energy.
Deprives the brain of fuel. What are the symptoms?
The symptoms progress depending on how low the blood glucose falls. The source describes a typical pattern. At moderately low levels, maybe 50, 70 mg, 100 milliliters, the CNS becomes hyperexcitable. This can cause symptoms like nervousness, trembling, sweating, and sometimes even hallucinations.
The initial agitation. What happens if it drops further?
If blood glucose falls further into the range of perhaps 20, 50 mg, 100 milliliters, the effects become more severe. Clonic seizures can occur, followed by loss of consciousness.
Seizures and unconsciousness. And if it goes even lower?
At extremely low levels, the seizures may cease and the person lapses into a deep coma.
Very dangerous progression. How is this clinically distinguished from a diabetic coma caused by ketoacidosis? They both involve coma.
That's a critical distinction to make in an emergency. The main clues are based on the underlying cause. Diabetic coma due to ketoacidosis involves hyperglycemia, very high blood sugar, metabolic acidosis, the likely presence of acetone breath, and often rapid, deep breathing, small respiration, as the body tries to blow off excess acid, CO2. In contrast, hypoglycemic coma due to insulin excess involves hypoglycemia, very low blood sugar, no acidosis, no acetone breath, and breathing may be normal or even shallow.
Key differences to look for. How is insulin shock treated?
The treatment is immediate administration of glucose. Usually this is given intravenously for a rapid effect. This quickly raises blood glucose levels and can often restore consciousness within minutes. Alternatively, an injection of glucagon can be given, which stimulates the liver to release its stored glycogen, also raising blood glucose rapidly. Epinephrine could do this too, but less effectively.
Get glucose levels up fast.
Urgently. The source emphasizes that prompt treatment is crucial because prolonged severe hypoglycemia can cause permanent damage to neurons in the central nervous system.
It truly highlights the critical nature of maintaining that precise metabolic balance. Too much insulin or too little insulin, and the system, particularly the brain, is put in serious danger.
Absolutely. It really underscores the elegance, the complexity, and the absolute necessity of the intricate regulatory systems involving insulin and glucagon that we've explored today.
We have taken a truly deep dive today, really getting into the source material, unpacking the intricate choreography between insulin and glucagon, all orchestrated by the pancreas.
We've seen its absolutely vital role in maintaining metabolic homeostasis, particularly that tight control of blood glucose.
And we've explored in detail the profound consequences when this delicate balance is disrupted, leading to the complexities of diabetes mellitus in its different forms.
By exploring the anatomy, the chemistry, the cellular mechanisms of action, the regulation, the crucial counter-regulatory roles, the pathophysiology of disease states, and the diagnostic and treatment approaches described in the material.
Our goal was really to help you, the listener, cut through the noise and truly grasp this topic.
To provide the clarity and depth needed to master this information, to understand this vital system like an elite professional in the field.
We hope this deep dive has achieved that, giving you a solid foundation.
So, to leave you with a final thought, something to ponder, building on what we've covered today, considering those remarkable and often very rapid metabolic benefits seen after bariatric surgery, benefits that seem to go beyond just simple weight loss. What other, perhaps still undiscovered, signals or pathways might the gut or maybe other organs be using to influence insulin sensitivity and glucose metabolism?
Could unlocking those secrets, those unexplored signals, be the key to developing entirely new therapeutic breakthroughs for diabetes and obesity in the future?
Or perhaps thinking about it from another angle. How might our ever-deepening understanding of the specific molecular cascades we touched upon today, the intricacies of the insulin receptor signaling pathway, or the nuances of the glucagon ChemMP cascade, how might that knowledge be leveraged to develop even more targeted, more precise, and ultimately more effective treatments for diabetes and other metabolic disorders down the line?
Lots to think about. Thank you for joining us on this deep dive into the fascinating and critical world of insulin and glucagon.
Until next time.